Solar energy is trapped and used to generate ATP and NADPH. First, the pigments, chlorophyll, within the thylakoid absorb light. Chlorophyll is arranged in thylakoid membranes in clusters called photosystems. There are two types of photosystems, PS1 and PS2. They are not named in the order they happen. Photosystem 2 comes before photosystem 1. Each photosystem is made up of chlorophyll molecules. Also present is a molecule that accepts electrons, called an electron acceptor. All the pigment molecules in each photosystem can absorb light energy in various wavelengths. The pigment molecules always pass the energy onto the reaction center, specialized electrons accepting chlorophyll. Once the reaction center has received the energy, the electron is excited. The electron is then passed on to an electron accepting molecule. The reaction center in photosystem 1 is now missing an electron and must be replaced before it can absorb more light energy. The source of the new electron is a water molecule. A water molecule is split in a series of reactions that release electrons, hydrogen ions, and oxygen atoms. Oxygen produced by plants comes from these water-splitting reactions. From the electron acceptor, the energized electron is transferred along a series of electron-carrying molecules, electron transport system. With each electron transfer, a small amount of energy is released. This energy is then used to move hydrogen ions from the stroma along with the thylakoid membrane and into the area within the thylakoid called the thylakoid space. And this same energy is released as an electron is passed through electron transport system, is stored temporarily in the hydrogen concentration gradient along the thylakoid membrane. There are many more hydrogen ions within the thylakoid compared to within the stroma. Therefore, the hydrogen ions move from an area of low concentration to an area of high concentration. While step 1 and step 2 are taking place, light energy is absorbed by photosystem 1. Once again, the light electron is transferred to a reaction center. The excited electron is moved to a high-energy electron acceptor. In photosystem 1, the lost electron is replaced by an electron that has reached the end of the electron transfer from photosystem 2. The electron that is received by the electron acceptor from photosystem 1 is used to reduce NADP, a high-energy compound, to form NADPH, essentially just adding hydrogen. NADPH is used to reduce carbon dioxide to make glucose.
Hydrogen ions from photolysis are forced from the stroma to the thylakoid space, but they cannot diffuse back across the membrane because the membrane is impermeable to these charged particles. ATP synthase, a mechanism that bonds a free phosphate group to an ADP molecule to form ATP, is within the thylakoid membrane to help the molecules move back across the membrane. The thylakoid membrane has the only pathway for the hydrogen ions to move down their concentration gradient. As the hydrogen ions move down their concentration gradient, they pass through ATP synthase. The ATP molecules are generated by the energy of the gradient. This is called chemiosmosis. This process is found again in the making of ATP in the mitochondria.
When there's enough NADPH and ATP in the stroma, the energy can be used to create glucose in the presence or absence of light. This is light independence. The series of reactions by which carbohydrates are created is called the Calvin-Benson cycle. Step 1. Fixing carbon dioxide. Carbon atoms in carbon dioxide is chemically bonded to a pre-existing molecule in the stroma. Molecule is ribulose biphosphate. The 6-carbon compound is unstable and immediately breaks down into 3 identical 3-carbon compounds. First stable products of the process. Plants that use this process are called C3 plants. Step 2, reduction. The newly formed 3-carbon compound is in a low energy state. To convert to a high energy state, they are first activated in by ATP and then reduced by NADPH. The result is PGAL, glyhydride 3-phosphate. In their reduced high-energy state, some of the PGAL molecules leave the cycle and may be used in glucose. Remaining move to the 3rd stage. Step 3. Replacing RUBP, the reduced PGAL molecules are used to make more RUBP. The energy supplied by ATP is needed to break and reform the bonds to make 5-carbon RUBP from PGAL. The Calvin cycle must repeat 6 times in order to create 1 molecule glucose. Of the 12 PGAL molecules that are produced, 10 are used to regenerate RUBP and 2 are used to make glucose. Cellular respiration releases energy by oxidizing glucose to carbon dioxide. Glycolysis, which means breaking up of glucose, is the first step in cellular respiration. Glycolysis breaks up the glucose from a 6-carbon molecule to a 3-carbon molecule called pyruvate using 2 ATP. This is an anaerobic process. No oxygen required. Glucose starts with 2 ATPs and makes 4 ATPs. The first stage of glycolysis, 2 molecules of ATP are consumed for each mole of glucose. Glucose is converted to fructose 1,6-biphosphate. Glucose is trapped inside the cell, at the same time converted to an unstable form that can be split into 3 carbon units. In stage 2, fructose 1,6-biphosphate is split into 2 3-carbon units of glyceride 3-phosphate. Stage 3, 4 moles of ATP and 2 moles of NADH are gained from each mole of glucose. Glyceride 3-phosphate is oxidized to create 2 pyruvates, the 3-carbon molecule. If there is not a sufficient amount of oxygen, the pyruvates must proceed through fermentation.
Go.
Fermentation, a way of reducing pyruvates to other compounds through the oxidation of NAD+ from NADH. It is a much less efficient way of supplying energy because it can only produce the amount of ATP that is generated in glycolysis. There are 2 types of fermentation: lactate fermentation and ethanol fermentation. Lactate fermentation uses NADH to convert pyruvate to lactic acid. The resulting NAD+ is recycled to continue the process. This type of fermentation takes place in single-celled organisms and some animal cells when they are temporarily out of oxygen. Ethanol fermentation, the second type of fermentation, a pyruvate is converted to a 2-carbon molecule and then into ethanol. This type of fermentation takes place in anaerobic organisms such as yeast and bacteria.
This is the Krebs cycle preparation. The pyruvates are oxidized and creates acetyl-CoA, essentially just a 2-carbon compound, and NADH. The acetyl-CoA gets transferred to the Krebs cycle and merges with the oxalic acid. Together they make citric acid. The Krebs cycle. The 2-carbon compound is added to the Krebs cycle and the 2 carbon atoms are fully oxidized to carbon dioxide. The energy released when the carbon becomes CO2 is transformed into reducing power in the form of NADH and FADH2. Many high-energy hydrogen pairs have been taken off glucose fluke fragments in the Krebs cycle and sent to NAD and the electron transport chain.
Go.
The electron transport chain. The components of the chain are protein pumps and carriers in the intermitochondrial membrane. High-energy NADH have been produced in the following steps when NAD is reduced into NADH. NADH then releases a hydrogen ion into the matrix of the mitochondria. 2 high-energy electrons enter the electron transport chain. When NADH molecules first enter the complex, hydrogen protons and high-energy electrons are split away, leaving NAD. The high-energy electrons are passed down the chain from one complex to the next. Each complex releases some energy from these high-energy electrons and uses it to power the active transport of hydrogen protons from the mitochondria to the intermembranous space. This builds an electrochemical gradient between the matrix and the intermembranous space. The energy in the gradient will be used to produce ATP during chemoosmosis. Chemoosmosis in the mitochondria couples the movement of the hydrogen ions down their concentration gradient to the synthesis of ADP to ATP. When the 2 last electrons emerge from the last complex embedded in the inner mitochondrial membrane, in the electron transfer chain at low energy, they join two hydrogen ions that will be released from ATP synthase after chemoosmosis. Oxygen is a reactant for cellular respiration and therefore combines with electrons and H+ ions, forming water as a waste product. At the end of cellular respiration, 36 ATPs are created.
What we do differently is we would use a bigger scale and make the model more 3D. The differences between our model and the real process is that the electrons in our model are not shown, and that the process in the real model is a lot more complex compared to the model we have developed. Obviously, the real model is not made of Play-Doh, and another point is there are many more parts in the plant that are not shown in our model. It was hard to show each process in detail, as well as being difficult to find materials that would stick to our board while also being creative.